Evening viewer, welcome to our Globe TV. Here's a breaking news. Yes, a research study conducted by researchers at the University of Cologne has set light on how nerve cells, that means neurons that regulate the movement of leg muscles in stick insects, operate in a rhythmic manner. The team discovered that the neurons responsible for triggering the deep pressure muscle in the leg exhibit a rhythmic activation pattern which is distinct from the activation patterns of neurons associated with other leg muscles. So far, it has been assumed that all of these so-called motor neurons are activated in the same way by central neuron networks. The study was published under the title "The Synaptic Drive of Central Pattern Generating Networks to Leg Motor Neurons of a Walking Insect is Motor Neuron Pools Specific" in the journal Current Biology. The USC research teams investigates the neural foundations of motion generation in animals, in particular those underlying locomotor activities such as walking. For these purposes, the team led by Professor Dr. Ansgar Bussege's analysis insects, among other arguments, as the requirements for the nervous systems regarding the generation and control of walking movements are very similar across the animal kingdom. In many animals, for example, there are networks in the central nervous systems that form the basis for the generation of rhythmic activity patterns for many forms of movements, whether for rhythmic locomotor activities such as running, swimming, crawling and flying, or for vegetative functions such as breathing. These highly specialized networks are referred to as central pattern generators. They generate the rhythmic motor activity of the muscles for movement in interaction with information from sensory organs. Neurons called proprioceptors proprioceptors report movements that inform the central nervous system. In the case of walking, they are located on the insect's leg. The networks do this by activating the so-called motor neurons that innervate the muscles. So far, it was assumed that such CPGs have the same influence on all motor neurons they target. In their new study, Angelina Rothi, Dr. Charlampos Mantijarius and Professor Busges also disproved these assumptions about the locomotor activity in insect. In their experiment, the scientists pharmacologically activated the CPGs in the central nervous system of the stick insect Carausianus morosus and investigated their influence on the motor neurons that innervate its leg muscles. They found that all motor neuron groups of the leg muscles except one receive identical drive from the network's rhythmic inhibitory signals from the CPGs. Only the motor neurons which innervate the depression muscle of the leg are controlled by the phasic excitatory drive. Interestingly, the leg depressor muscle is precisely the muscle of the insect that is responsible for generating leg stance during any walking situation, regardless of whether the animal runs up or down horizontally on the ceiling or on a branch. The rhythmic excitation and thus the specific activation of this motor neuron pool by the CPGs could serve to ensure the exact timing of the contraction of the depressor muscle and thus the start of the stance phase and its stabilization, explained Professor Busches. BNS report, Globe TV, Delhi, Bharat.